Hi, my name is Lydia Son. I'm in the Mechanical Engineering Department at UC Berkeley. Today, I'd like to talk to you about high-throughput DNA-directed patterning to recapitulate biological signaling scenarios. The adult stem cell reside in very dynamic niches. And in fact, stem cells can be found within the epidermis and hair follicles of the skin. They can be found in the crypts of the base of the crypts of the adult intestine, as well as the bone marrow, and finally in the subgranular zone of the dentate gyrus of the hippocampus. Now, adult stem niches are highly complex, and the stem cells receive a diverse spectrum of cues. And these cues consists of, for instance, cell-to-cell communication, justacrine signaling through cell-mediated contact between the cells, as well as soluble cues that are released for paracrine signaling. As well, the ECM, the extracellular matrix, can provide cues in terms of its composition and sequester cues that can form a neural stem cell and form a stem cell. And finally, biophysical cues, for instance, the matrix stiffness, the topography of the ECM, this can inform as well the stem cell. And it's really all these cues, the biochemical and the biophysical cues, that really coordinate temporally and spatially to instruct stem cell fate decisions. And these decisions are, as I've listed here, differentiation, programming, cell cycle entry, apoptosis, proliferation, quiescence, and migration. If one were to study these dynamic signaling networks, and in particular neural stem cell niche, how can we do this? We would really need to have a method that could really give us this ability to have spatial control and temporal control of all these different signaling networks in order to study a particular niche. And for our case, it's the neural stem cell niche. Now, what I'd like to describe for the rest of my talk is one particular method that we've developed, which we call high-throughput DNA-directed patterning. This is work of a collaboration that I have with David Schaefer in the Chemical and Biomolecular Engineering Department at UC Berkeley, and this is the work of our graduate student, Olivia Scheidler. Now, before I get into high-throughput DNA-directed patterning, I first have to tell you what DNA-directed patterning is. And really, this is a really neat technology that was developed by Dave Schaefer and Zev Gartner, in which what you could do is really pattern cells using DNA hybridization. Now, how does that work? Essentially, what you have is you have an aldehyde-coated glass slide, and you can conjugate a DNA molecule or single-stranded DNA oligo onto the surface of this glass slide through reductive amination. You can take the complementary strand of that pattern oligo and tag a cell, in this case a neural stem cell, with that complementary strand. And once you flow your solution of tagged cells on top of your pattern substrate, hybridization will occur, and you essentially capture your stem cell or your cell on the pattern with the patterned oligo. Now, you can use different sequences to capture different cell types. So for example, what you see here is we've captured, as in the schematic, is that a neural stem cell is captured with one particular sequence of oligos. And here's an astrocyte that's been captured by a different sequence of oligos. And essentially, you can have orthogonal DNA strands that coordinate the assembly of different cell types onto a substrate. Now, DNA-directed patterning is quite revolutionary, given its simplicity. Again, it all rests upon DNA hybridization. But the patterning at the time was done with a nanoenabler. A nanoenabler is really nothing more than an atomic force microscope probe. And essentially what happens is that you dip the probe into your solution of oligos, and then you spot your substrate with using the nanoenabler probe. Now, this method is serial and slow. You can think of it as that what you're doing is really patterning your substrate pixel by pixel. And as you imagine, this is a very slow process. As well, patterning is confined to a very small area. You certainly couldn't pattern a 3-inch wafer, and it really was. It's really limited to one or two cell types, not because of the actual process of hybridization, but more the fact that alignment with the nano enabler to align different oligos, different sequence or strands of oligos onto a substrate is very difficult with the nano enabler. And finally, if one were want to pattern solid phase cues that you would find in, for instance, in a neural stem cell niche, it would be very difficult to do merely because the sheer numbers of having that many solid phase cues on your substrate. So the real question that we sort of settled on was really, how do you fabricate efficiently and over large areas instructive surface DNA patterns that could accommodate cell types and ligands, these very cell types and ligands that you would find in the neural stem cell niche? Well, this is where the expertise of my lab comes in. We're experts in lithography, different lithographic techniques. And what we realized is that you could do multi-layer photolithography to do your DNA-directed patterning. And in this way, instead of exposing and patterning pixel by pixel, you could then pattern really arrays of different areas of oligos all on a large substrate. And in this way, it's high throughput because really it's a parallel process. Now, how does high throughput DNA directed patterning actually work? It essentially is a layered method whereby the first layer, what you do is you spin your resist onto a substrate and you expose the pattern that you want to, you expose the pattern of a mask onto your substrate. And through development of the resist, you essentially expose areas of the aldehyde coated glass, and thereby you can guide the surface conjugation of a particular DNA oligo onto your substrate. You can strip the resist and then repattern again, put down your photoresist, and then expose another pattern onto your substrate. And this time, use a different set of oligos. They may be the same oligo sequence, but you're just patterning different areas, or you may be patterning a entirely different oligo sequence. Again, you can strip off the resist, apply more resist, and then do another exposure, and this time be able to conjugate a different oligos onto your substrate. Strip the resist, and then essentially what you're left with is a pattern that's multi-layered and that has different oligos patterned onto a substrate. And to confirm that you've successfully patterned your substrate, what you see here is what we've done, which is essentially take the complementary oligo strands, each one labeled with a different fluorophore, and perform hybridization. And essentially what you see here is how well we're able to have tight spatial control over different orthogonal DNA strands. And you can see here, this is the space bar of 500 microns. And in fact, this was a pattern that we repeated on a 3x1 glass slide, done all in parallel. This would have been extremely difficult with the nano enabler. The question really is how many layers can you actually pattern? How many different oligos can you pattern onto your substrate? Well, we've done a test, and what we found is that remarkably, we can pattern up to 10 different DNA layers on a substrate. Here, what you see is what Olivia had done, which was she had patterned her first layer of oligos, then she patterned the next layer, and so forth, all the way up to 10 layers. And what you can see is that the very first oligos that she patterned, they were not affected by all these different patterning steps. What I want to really emphasize is really how remarkable this actually is. The fact that the DNA oligos have survived the entire lithography methodology. It survived the spin coating of the photoresist. It survived high baking temperatures whereby we polymerize the photoresist. It survived the acetone stripping that we do to take off the photoresist and then reapply the photoresist. And finally, it survived the photoresist developer itself, which has a pH of greater than 12. Often it's 14. So it's a very caustic developer, and yet the oligos have survived. Now, with high throughput DNA patterning, you can achieve spatial organization of the cells. And for instance, here is an example whereby what we did was we used a single oligo sequence and we patterned neural stem cells that we tagged with a fluorophore. And you can see how well we're able to pattern these cells into the logo of the UC Berkeley and of Cal. Okay. We could also pattern neurostem cells using 3 different sequenced oligos. Essentially, we took our neurostem cells and we divided them up into 3 different populations. Each population was dyed with a particular cell tracker dye. And you can see how well we're able to pattern these 3 different neural stem cells, or 3 differently stained neural stem cells, using the 3 different sequenced oligos. Now, to flex our muscles somewhat, but really to show you how flexible our method is, let me just show you that we can pattern other cells. This is where we've patterned HUVEC cells, and essentially we were trying to recapitulate the tissue architecture found in the intestine. And then this is a case where we used our high throughput DNA directed patterning to assemble in vitro tissue models. In this different cell types that we pattern. The yellow corresponds to MCF7, or malignant breast epithelial cells. The blue corresponds to HUVEC cells. Those correspond to we want those to correspond to the vasculature. The MCF10As are the normal breast epithelial cells. And finally, the white correspond to invasive MDA-MB231 malignant epithelial cells. This is just a mock-up, but it really shows how much control we have over patterning different cell types and what you really could do with high throughput DNA directed patterning. Now, by essentially patterning different feature sizes on your substrate, you can really tune the number of captured cells. So here is just an example, and this really shouldn't come as a surprise, but it was a nice thing to show. Here is where we have a circle size that is a diameter of 30 microns all the way up to 120 microns. And what you can see here is that essentially we can control the number of cells based upon the spot size that we pattern. We can also control the intracellular distance. Here's an example whereby we have the control. And again, I want to emphasize this is all done in parallel. We're patterning all these different features. Now, beyond patterning cells, we can also pattern solid phase ligands. We can attach an DNA oligo onto a ligand through click chemistry, and then we can perform our usual patterning method. And so here's an example where we took green fluorescent protein, attached an oligo sequence onto it, and then we also tagged an M-cherry. And through our various patterning, we patterned different features of M-cherry and green fluorescent protein. And you can see how well we have control. This is just a really nice example to show by controlling the concentration of our ligand, we can control the number of ligands that we actually pattern. And so here we vary the concentration of the green fluorescent protein in each one of these patterns. And again, you can see how well we're able to pattern with great control of the ligands. So now that we have our toolbox of the ability to pattern spatially cells, different cell types, and ligands, what could we really do? Well, our whole goal is really to look at and recapitulate the neural stem cell niche. one of the simple things that we thought that we could do with our patterning technique to show that it works and really that you can get meaningful information was really to see what happens to a neural stem cell when it's presented with two competing cues. And the two competing cues that we chose is FGF2, which promotes proliferation, and Ephrin B2, which promotes differentiation. And we really want to know what happens when this neural stem cell sees both. What will it choose? Will it choose one over the other, or will it just essentially, as a joke, to remain confused? So what I show you next really highlights the power of high throughput DNA-directed patterning. This is essentially just an image of a small portion of our chip. And what you see is it consists of many small squares. Each one of these squares is actually a particular culture condition or a culture condition that we're testing. And you can see that we've essentially been able, and this is, again, I want to emphasize, it's all done in parallel. We've been able to create thousands of culture conditions on a single chip. And what we're interested in, again, was to look at these competing cues. And what you see here is what we chose to do is to look at three different spatial presentations. One in which the neural stem cell pattern in the center is essentially first sees just FGF2. And then surrounding that is Ephrin B2. And the question really was, would the neural stem cell just remain in FGF2 or would it actually see Ephrin B2? And the flip side would be, what if it actually starts off in Ephrin B2? Would it stay there or would it extend out into FGF2? And finally, in this pattern, it's really to see, well, what happens if it has equal opportunity to either go to FGF2 or Ephrin B2? Which one would it choose? And here you can see is just in the bright field image are the neural stem cells that we were able to pattern in each one of these black areas right here. Again, I just want to emphasize the ability that we were able to use this high throughput DNA-directed patterning to essentially create these different culture conditions, to repeat them for statistical rigor, and to perform these all in parallel. This is something that simply cannot be done by any other patterning method currently available today. Now, what Olivia had done was to essentially take images every hour of these neural stem cells over a course of 4 days. And this is just a combined picture or image of these neural stem cells. You can see that they're extremely active. They're dividing. They're feeling their entire environment. And I think that is rather remarkable that even though they start off in either the FGF or in the Ephrin B2, they are exploring their entire region. Okay, they don't extend beyond these squares right here. We use PEG to confine the cells within a particular culture area, and essentially you also see that the cells are dividing with one another. Now, after 4 days, we fix the cells and then stain them for different markers. And what we found either one, we found that the neural stem cells preferred FGF. They prefer to be in this area, and they prefer to essentially proliferate. Okay, and this was quite a surprise, but it was really nice to see. So while we found that most neural stem cells prefer to be in FGF2 and proliferate, we did find a small subpopulation that, while preferring to be in FGF2 and therefore should proliferate, they actually ended up differentiating. And this was a really surprise for us, and it goes against what you would expect to see. So the question really is why? Well, we believe that, as you see in these arrows, that even though the cell body stays in FGF2, their neurites do extend into the Ephrin B2 region. And perhaps there is communication from their neurite to the main part of the neural stem cell that essentially tells it to differentiate. We're investigating this some more, doing further experiments, but it is really an amazing phenomena that we found, and this can only have happened because that we were able to look at thousands of culture conditions at the simultaneously, that we were able to find this small subpopulation. Now, as I mentioned at the very beginning of my talk, within a stem cell niche, in the neural stem cell niche, there are multiple cues that coordinate temporally and spatially to instruct stem cell fate decisions. And what I've just spent the majority of my time right now is to talk about how we were able to spatially instruct and recreate spatially pattern different cell types and ligands to create a niche. So what can we do temporally? Well, this is just how can we achieve this temporally through high throughput DNA patterning. And what I'm about to just show you is a little bit of a teaser. We're still working on it, but essentially we can use DNase to essentially remove the ligand presentation from our patterned niche on our substrate, and in this way we can induce temporal control over our ligands. So in summary, what I've shown you is a really flexible, precise, and efficient technique, high throughput DNA directed patterning. It's using very simple photolithography and multi-layer photolithography to create a variety of different patterns. We're able to use essentially DNA hybridization to pattern our cells and our ligands, and the ability to essentially just be able to have that flexibility to use different cells, to pattern different cells, different ligands, to be able to pattern from microns to millimeters, all in parallel. It really is quite a remarkable and flexible technique. With that, I'd like to acknowledge my group, and in particular, Olivia Scheidler, who was really the main driver of this entire project. She did an amazing job. She graduated and is now a lead scientist at ADDIMAB in New Hampshire. Molly Konsminski is my postdoc, and she's now taken over this project and really pushing high throughput DNA patterning to the next level. And of course, I would be remiss to not acknowledge my collaborator, Dave Schaefer. He's just an amazing, wonderful, creative scientist, and I really enjoy working with him. And I also like to acknowledge my funding, the NIH Berkeley Stem Cell Center and Michael and Margaret Cheka. Thank you very much. And now I'd like to take questions.